things again. I was saying to Patrick just before, it's amazing because in daily life I often have the feeling that there is not progress and everything is going so slow. And then I look back, okay, what happened in the last 2 years? And actually there were quite a few things that I can talk about. So the title I gave here is what I sent to Patrick in December or something. It was it's basically the title of a paper that we have just submitted, and now we're waiting reviews. And I will get to that topic, so it's ape in the wild, towards the end of the presentation. But will before that spend a bit of time, giving you some updates, but also in the beginning reminding you of what actually this work is that we are doing, because we don't get to talk about that too often. So I think it's good to have a reminder. So I looked that up 2 years ago. Yeah, that's here. So in the UBC seminar, so I had a talk titled Automated Workflow Composition in Mass Spectrometry-Based Proteomics. And basically that was based on a paper that we had published half a year earlier in bioinformatics called, of exactly that title actually. And that was joint work with Magnus Palmblad, who is a microbiologist and proteomics researcher in Leiden, the University Medical Center. John Eisen, a bioinformatician, very active in the Elixir network, who is the one who initiated, for example, the EDAM ontology and the bio.tools registry. And Veit Schwemmel is a biostatistician at the moment working at the University of Southern Denmark. Nice collaboration where we actually just try to create proteomics workflows automatically. And I will talk a bit more about what that means in the following. But that was a work from, published in 2018, but we worked on that, let's say, from 2 years earlier, roughly that. To get before we start, some of the terminology clear. So what do I mean when I talk about scientific workflows? What I mean by that is also what some people call computational pipelines. Many of you probably are using them or building them on your own. So what I mean by that is computer programs for data analysis mostly that are characterized by being mostly orchestrations of existing tools. So collections of tools, you need libraries, you take tools from here and there, and the program that you write is more the kind of glue code between them that defines how data is passed from one tool to the other, but it's not about implementing algorithms so much. It's more about putting existing pieces together to achieve a certain goal. And there's different workflow environments. Like on the left, I've put a screenshot taken from the Workflow Hub, which is a new platform for sharing workflows that is there provided in CWL, so the common workflow language, and that can be visualized in a style like this. So people who have worked with bioinformatics workflows before might recognize this look and feel from Taverna, which was one of the first bioinformatics workflow systems. But if you look at this definition of works a bit more abstract, it also means you can in the same way implement a workflow in a scripting language, or just as an R script, for example. And it has the same properties of essentially its code that orchestrates the calls to different other tools to achieve a certain goal. And I'm not completely sure which of the other workflow platforms that are around you guys are currently using here, but just to name a few that are popular at the moment, there's a Galaxy platform, there's Nextflow. I know that people are using SnakeMake, which is a bit of a Python-style workflow environment. There's Riddle, or the workflow description language, and many more. And there's also trends that come in waves, but the principal pattern is always the notion of the workflow in all of these are the same. And I'm going to abstract a bit because our work is not about any of these in specific or in particulars, but we're trying to abstract a bit and develop technology that makes the construction of these workflows easier in general. So that only as a short reminder what these workflows are that I'm talking about, and recap from the talk of two years ago. What I also sketched there is the vision that we're working towards. And I usually describe this as working towards a route planner for scientific workflows. The analogy being yeah, everyone is familiar with Google Maps as a route planner, for example, and what people typically do there is saying something like, okay, I am here, I want to go there, maybe I have some ideas on how I want to get there, for example, about public transport or not, and then Google Maps would come back with a number of suggestions for routes I could take. And the idea is, couldn't we have something like that also for scientific workflows, where we say to a system just, okay, this is the kind of data that I have. I want to have this other kind of data. I might have some ideas about the operations that I want to use on the way, or some other things that should happen in this workflow. And then the system comes back to me with suggestions on how I could achieve that goal and which workflows I could use. And then I would pick one, and just that gets executed. So, I mean, we're not there yet, and it's quite some way to go, but we're still working towards that, and we're making progress. But don't expect they're ready to use systems at the end of this talk. I'd rather talk about a bit what the current state of the art is, what we can do. So the updates compared to 2 years ago are basically the following, and I'm going to talk about all of these four in the following a bit. So one thing that happened last year, really just before the pandemic kicked in and we started working from home, was that we had a Lawrence Center workshop on this topic, automated workflow composition in the life sciences, in Leiden, with like, I think 36 people from 10 countries who all more or less work towards a vision like that. I mean, with different focus points and focus areas, but sharing the same idea and wanting to explore if that is at all possible and what we can do and where we are. So in June last year, so summer last year, we released a new tool that can actually do some of the exploration, and I'm going to show you a bit like what we can do with that later on. In autumn, we started to develop a nice like web interface for that, because the original tool is more like a command line and Java interface that is not so convenient to use for everybody, and also meant more for developers and not so for end users, but the web interface then is. And yeah, the latest thing is this mentioned study that we say we use this tool in the wild, so on the bio.tools registry, to see how it behaves when we really release it to the real world and go away from the toyish examples that we had before. So that's more or less a plan for today. Yeah, briefly, this Lawrence Center workshop last year, and I dug out these pictures, and it seems so unreal that there was a time where we could really meet with so many people in one room. It was a bit bizarre that week, because as you can see in the poster, it's really that week leading up to 13th March, and in the middle of the week, like the American participants left us overnight because they were worried of not allowed to get in again, and these kind of things. So it was a bit bizarre, but we could carry out this workshop and bring it to an end, and it was quite a good experience. We're still finishing up writing the workshop report, which was also delayed due to people having so many other things to do during the pandemic. But we expect to submit this to F1000 Research early March. So maybe stay tuned for that if you're interested to read more about the big picture of this area of work. One thing that is a bit of an outcome of this workshop is this picture of the workflow life cycle, where we, after a week of discussions, tried to summarize, okay, what is generally the process of creating a scientific, in that sense, like life science or bioinformatics workflow. And I don't want to talk about this picture too long, because it's really a bit of the summary of a whole week, and we could talk about many aspects of that in quite a long while. But what you can see here in principle, that we distinguish two principal roles. So on the one hand side, the workflow user in the life science field, that would mean mostly the biologist, and on the other side, the bioinformatician as a workflow developer. Of course, sometimes you have two in one. Some people are bioinformaticians and biologists, according to definition, and would do everything. But we do see the role of a professional workflow engineer also emerging. So it makes kind of sense to separate these two perspectives on workflows conceptually. Yeah, so the application side on the left is then a bit more concerned with really the scientific questions, with generating the hypothesis, applying the workflows to data. But from this point of the cycle here, people are actually happy to take a workflow that is provided, tried and tested, that where it can be assumed that it will be produce reliable scientific results and that then can be used in the scientific process. Whereas the other side is more concerned with starting from some question or hypothesis. How do you actually get to a workflow that is good enough for being used by practitioners or in the lab to answer those questions? Is roughly the distinction. And yeah, the phase that we are focusing on today is mostly the first part of the cycle here, if you want to say that this is the start. So it's mostly the exploration and composition. So the early phases where you wonder, okay, what kind of workflow conceptually could actually solve my problem, without getting into too much of the technical details, but looking at, okay, what is the method and what tools are available. So that will be a bit the focus of today. And that is called what we call exploration and composition. So the problem there is, and some people of you who have built workflows before might use that. So the basic problem is that there are thousands of computational tools nowadays, and yeah, combinatorially, you can say there's even more possible workflows that you can build with them. And what many people do, after a while in the field, you would know certain tools, you know what the good tools are, what your favorite tools are, so you put them together, but you never know if there are others, and how they could combine. And it can be a lot of work going through all the documentations and looking up type compatibilities, what tools can really work together. So that's a bit the challenge. I mean, to put this a bit in numbers, the bio.tools registry, so that's a big repository for bioinformatics tools run by Elixir, the European Life Science Network. Currently, it has more than 17,000 tools in that registry. So if you face an amount of tools like that, and you're supposed to find those that solve your problems in the best way, and even maybe the workflow that you can build from these problems that solve your problems in the best way, that can be a bit of a challenge. And of course, many people have experience and will just start with a few tools they know, and they know it's tried and tested and results, and go from there. Well, we could also see, okay, if you have no idea what the tools are, but you have a kind of problem, how to best, in that case, find the combination of tools for a specific problem. Another point here is that always new tools are emerging. So if you want to make sure that you're using the latest tools for the workflow that you're building, how would you do that in the light of so many tools available? And that leads us a bit to our research question from more the computer science side. So this is not the biologically research question, but really more like a meta or computer science question. That is, how do we find the best combination of tools for a specific problem? Where the first question here would be what best actually means. So even that is not clear, or at least has many ways to fill it in. And then, the connected question to that is, once we know that a bit or have an idea of how to do that, to which extent can such an exploration process be automated? So that not a human user has to go through all the tools and study the documentation. So that's what we're trying to do. So I mentioned we have built a tool for that. So it's called APE, or the Automated Pipeline Explorer. And I think it's also a nice depiction of what it does, because I mean, APE it's an intelligent animal in a sense, and it does also something that is not trivial, but certainly like too tedious for a human to do. So it's great if the APE can help there. So APE is our, we say, toolkit for automated exploration of scientific workflows, and that is what I mentioned before. We provide as a command line interface and Java library. So that is a bit meant for being integrated into other workflow frameworks that say, okay, for example, Galaxy, you could say, if you want to explore possible workflows at some point, that you want to build a Galaxy that could call APE as a plugin and provide the Galaxy user with suggestions for workflows. So that's a bit of the idea, but APE itself is not tied to any workflow framework. some terms that are relevant for when working with Ape. So the one here is called the domain model. By domain model, we mean, yeah, the knowledge base that Ape needs, or the formalization of all the technical knowledge about the application domain. So which tools are there? What are the inputs, outputs? What are generally the data types that we have at our disposal? All these kind of things. This needs to be provided in a machine readable way. That is the domain model. Then we have a second input, the workflow specification, and by that we mean that's a bit of like in Google the query or the search that you put into the route planner, where you say, this is my input, that is my intended output, these are the ideas I have on the way. And yeah, providing Ape with that, will give you usually a list of possible workflows for this specification. Maybe as an example that makes it a bit more clear. So it's again an example from proteomics. Basically this existed already in the last talk. So what we use as a domain model is these EDAM ontology. So that's the embraced data and methods ontology. I think it's the biggest technically oriented ontology in bioinformatics. I'll show a picture in a second. And the second part of the domain model here is, Elixir, the bio.tools as a platform, and we need tools with functional annotations, and that is illustrated here. So what is a functional annotation? Is in principle we say what a tool is doing. So we provide an operation term and describe what the operations is, and we say what the input and output of a tool would be. And so in this example, Comet is the tool, that's the tool name. It has as input mass spectra. The mass spectrometry spectra has been corrected. I think it's now mass spectra in MZ, ML, for example, in other formats. Its output is a peptide identification in a other ML format. The operation term is given as this. So that's meant by functional annotation, and we do have them in bio.tools, or bio.tools enables to provide these functional annotations. Not all tools in the platform do, but those that do, we can also use as input for Ape or as part of our domain model. Here's an excerpt of what EDAM is. So this ontology as controlled vocabulary, basically it's a big tree. I need directed acyclic graph, actually, but you can imagine it as a tree that has different hierarchies and categories that defines the vocabulary that we can use. So tool annotations in bio.tools can only use terms that have been defined in this EDAM ontology. And the operation, for example, could be, okay, this is an operation term, that can be a calculation. And if we use retention time prediction, we know from the context in the ontology that this is an operation, and this is a calculation is an operation, and so on and so forth. So in that, EDAM has parts for operations, but also for data formats and data types. The difference between formats and types is here, as you could see here. So the type is a bit of the semantic data type, where we say, okay, it's mass spectra. That's a bit the type of data that's in there. And the format is more about how it's represented technically, which kind of XML dialect, for example. So that's our domain model, knowledge base as input. And now we can put one request, for example, this workflow specification here that says, okay, I want to have a mass spectrum in Tamara format, and my desired output is an amino acid index, and there any format is fine, I don't care. And constraints, so additional requirements for on the use, I want to use the operation retention time prediction. So this request we can feed into Ape, and then it will come with, for example, workflows like this, but usually many more, but this should just indicate the kinds of workflows it would produce. I don't know if any of you works with these tools or the kinds of tools here, but these would be quite reasonable workflows that it would suggest. Yeah, and from here, The workflow developer can take these suggestions further, select some, implement them, execute to see how they really behave, benchmark them, and so forth. But APE is really focusing on facilitating this exploration phase. Yeah? So given a large collection of tools and a request, it will automatically generate suggestions of what workflows would solve the given problem. That is the idea. So for those who are a bit interested in the algorithm and how that works, so the intuition of that behind is that's not exactly the way it's implemented, but that gives a bit of an idea what the idea behind the algorithm is. So what would happen in the first phase of this algorithm would be that we create a search space. The idea is simple. So we start with the data that we have as input. So for example, this mass spectrum in temporal format, and then we just look, okay, if I have this kind of data, which operations can I apply? And when I apply those operations, what is the output of that? For example, here, if I do just a type conversion with MS Convert, I have the same kind of data, but in a different format. If I then do a plot on that, I get a different type of data in another format, and so on and so forth. And by that, we can span a huge tree of possible executions. It's really exponential growth, and that also explains why we have so many possibilities. But this is really much of what is technically possible according to the annotations that we have in bio.tools. So this is usually too much, but we have other information in the specification because we also define the goal type, so the output that we want to have, and we define additional constraints and kind of like operations that should be used. And that allows the algorithm to filter for certain paths and only follow those further that really satisfy the specification. And those that remain then will be our candidate solutions. Yeah, so this is an intuition how that works. It's not implemented directly in this way. This is a few for the more technically oriented or computer science oriented among you. So this is internally, this is a program synthesis algorithm based on a logic called semantic linear time logic, or SLTL. We implement this as an iterative deepening search. So that means we would increase the length of the solutions step by step, starting with workflows with one step only, then looking at workflows with 2 steps, then with 3, then with 4, rather than looking for very long workflows from the beginning. And the way it's done internally is that we encode all these information that we have, so the domain model, ontologies, annotations, workflow specifications, as the SAT instance. So SAT servers are satisfiability servers, and it means they're working with Boolean formulas in a controlled normal form, conjunctive normal form. And this is really, I mean, it looks ugly, but SAT servers love it. And so the SAT server then comes back with a satisfying assignment of variables, and that can be translated back into actual workflows. So that if anyone is interested in reading more about that, I would just refer you to a paper where we describe that. But for the talk today, that doesn't matter too much. As I said, brief mention of the APE web interface. I was kind of hoping that I could give you a short demo of that already today, but it's not completely possible because the student group working on that is finishing up this week. They have their end presentation on Wednesday and have to hand in their project on Thursday. So they are now really in their last phases and trying to fix the last bugs and finishing everything. So it's not super stable to use online, but maybe next week it can happen. So that's good. And definitely what we see already is much easier and friendlier to use in a command line interface or a Java interface even. It comes with, and that's also the main difference to the plain APE tool, it comes with a number of ready-to-use domains that are, they're pre-configured and that people can just use for exploring workflows and see what the tool is doing. Users can also create new domains in the web platform and share them with others. And finally, also workflows can be exported to CWL, so this common workflow language, which is emerging as a standard for workflow exchange, I would say. So maybe next time, or I will send an update through email if this really works. We would be interested in many people using it and trying it out and giving it feedback what we what we can do with it. Maybe a bit of background information. So these software projects is something that at the Department of Information and Computing Sciences, the computer science students do instead of an individual bachelor thesis. So they do a software project in groups of 10 to 12 students for a real client from outside who also has to pay a certain amount of money for that to the department. And then they really develop within one semester a software, usually prototype or product for that. That is what's happening. So this is why we know they're finishing this week. Quite nice. And yeah, we will take it over afterwards and work on it further, but they have really set a good basis for this platform. So if you have any development projects like that, maybe consider turning this into a software project at the department. Always looking for more projects. Okay, now so from the title, the ape in the wild. So this is again joint work with the previous authors of the paper, and the idea was basically the following. So far we applied ape and its predecessors always to relatively small domain models. Yeah, so the first bioinformatics paper, what we did is we selected a set of roughly 25 tools, and then we sat down and in a nice CSV table, we discussed the annotations for inputs and outputs. We had a few use cases for workflows that we fed into the tools, and then it kind of worked out nicely, but we were also always adapting the annotations so to produce better results, and it was a bit of really exploration and design phase. So yeah, but in the meantime, so since over the last 2-3 years, the bio.tools registry has really matured. On the one hand side, in terms of content, so there's many more tools available in the platform, but also more tools with good functional annotations. And it has also now a fully functional REST API, so that you can, from another computer program, fetch the tool annotations so that it can be processed in a machine readable way. That was also not really possible in the beginning. But now we have these two developments and these two features. And our question that's, okay, what would now happen if we take the same use cases as before, so the same workflow ideas as before, but now we use ape and the bio.tools directly. Yeah? And instead of having a small handcrafted set, we let the explorer directly work on the bio.tools registry. What would happen then? And that is the basis for this study here. So the setup for this kind of, let's call it experiments, but it's not the lab experiments that many of you are doing, obviously. We did the following. So we took again the 4 use cases from our initial paper. They all start with the mass spectra in term raw format, as we saw before, but they're aiming for different outputs. Yeah? So one is for the amino acid index, the other wants to get a pathway network, protein identification, and so on. And then in addition to this input-output specification, there's also always a list of constraints, of ideas, of what should happen on the way. As I said, so for example, I use peptide identification, we saw that before, but actually for this use case, there's 3 more operations that should be used, but also a constraint that says it specifies an operation that should not be used. And they're all more or less in the same style, that they formulate requirements on operations that must be used or exclude requirements that must not be used based on domain knowledge. Nevertheless, so we know from the previous study that we need these constraints to obtain good results, but still we explored workflows in the study in 2 modes, namely one with input and output only and without constraints, and the other mode is input and output with constraints, and just to see a bit what the difference are. And then we also had 3 different domain models for that study. One is, we call it the original tool set. It is not really exactly the original tool set, but basically the tools, but they're not in our own local repository, but on bio.tools, but corresponding to the small tools. That's the smallest set with just 21 tools. And just to, if there are some guys here around familiar with the field, these are just, I think these 21 tools that are in the list, just to give you an idea what kind of tools are involved. I will not show you the lists of tools for the other two domains. Just note so that the medium-sized domain is, we call it the proteomics domain. So there's a predefined domain or subset in bio.tools already for proteomics, where a group of proteomics researchers have gotten together to say, okay, we now define a list of proteomics tools in bio.tools, where we also make sure that they are of good quality and complete and cover all the important tools. And that tool set in the end was 271. And then, the big set, of course, is here then the full bio.tools domain model with almost like almost 1,700. And you might notice here now that, okay, this 1,642 is of course not the full 17,000 that I mentioned before as number of entries in bio.tools. The reason is simply that not all tools that are registered in bio.tools have the full functional annotations. And for APE, it only can make use of tools that do have annotations. So we discarded tools that are not well annotated already, so and that boils down to a smaller set. So it's only a fraction of the full bio.tools, but it's still a large amount. I mean, if you imagine to go through these tools as a human person, manually, just to figure out systematically what the best combinations are, that is quite a task that no one would want to do. Probably not even for the 271, for the 21 maybe. But doing that really systematically and exploring all possibilities is still a bit of work. Okay, so we ran these experiments with you, which you can calculate rather, like 4 times 2 times 3. So it's like the 24 in total. And what we did, we looked at 2 things. So once at the numbers, how many workflows do we get from a different perspective? And then we looked with a bit of expert knowledge and background knowledge at the workflows themselves. And I'm only, because it's a bit the same flavor of all the 4, I'm only going to look at the first one of the 4 workflows, of the 4 use cases here. Another thing to note, so we configured the search to stop after having found 100 results, with the reason just being, I mean, no one wants to look at more, and we needed to stop somewhere. So we just said it's 100. It's a bit arbitrary, but we found it sufficient to see effects. So what we report here is basically in the table, it's the number of workflows found for each of the experimental setups here. So the 3 domain models, and always like input and output only, or input and output and constraints. And then we report them per length. And that means length 1 would mean a workflow with just 1 step. This means, so in none of the cases we find a workflow that solves this problem with just 1 step. And the same for 2 steps. And the first solutions to this problem we found in a workflow here, length 3. So there's 1 workflow who can solve this problem with 3 steps. And there are 6 workflows who can solve it with 4. And in this setup, there's 1. So that's what we mean. And what we see, so if we are less strict and only use look for input output, usually we get more solutions, which is not completely surprising, because if we are stricter and we make more constraints and more requirements, we get less options. But the nice thing here in this case is that for all cases, we do actually find workflows at the end. And then also in effect, it's a bit as expected. You see that if we do not only take the original tool set with the 21 tools, but the extended one with the 270, we find more solutions per length. So in this case, for example, there's 12 instead of 1 for length 3, and 38 instead of 1 for length 4, and so on and so forth. And this is also not too surprising, you might say, because if I have many more tools available that do similar things to the ones that I have here, combinatorially, there are just more combinations that should be possible, and this seems to be indeed the case. when we look at the full tool set, we get again a few more, but not so many more, which is interesting. But this already, we take it as a good sign, we would say, because this is supposed to be the proteomics tools and bio.tools. And if I go beyond that, I do not find much more, meaning that probably this extended toolset does not miss very many of the relevant proteomics tools that are elsewhere in the registry. So that is a bit how we read it. And in fact, we do get similar results for the other 4 use cases. So I won't go through them here. Finally, to give you a bit of an impression of what these workflows are. So that is the second thing that we looked at as a part of the evaluation. But for this study, really the evaluation is a bit qualitative, really exploratory, that we just said, okay, with a bit of expert background knowledge, when we look at these workflows, what do we observe? So this is not a complete thorough analysis, but a first thing. So what can we see when we look at the workflow sets? And I don't show you all of the workflows, all of the 100%, but these on these slides here give more a bit of an impression of what the workflows are about. And for example, for the original tool set, so only exploring workflows with a set of 21 tools, we see that this closely resembles the results that we had in the previous study, including, in effect that we see here that tools and small workflows are often used redundantly. For example, you can see here that MS Convert, so the type conversion tool, is used 2 times after another, which is technically possible and not forbidden, but probably not very useful. Why would you convert a type 2 times in a row when you can do it 1 time usually does the job? And that is a fact we see more often as workflows get longer, they often contain tools or tool sequences that are not wrong, but also not very useful because they're just repetitive. So this is something we should look into in the future, how we can probably avoid that better. But it requires a bit of context understanding, so it's not so straightforward to teach that to the algorithm. So the extended tool set, so if we go to the 270 tools and many more, one of our domain experts noted there are indeed several new and sometimes surprising workflow suggestions, which could nevertheless work. So he said he would never have thought about that, but could be an interesting combination to explore. So maybe go that. And that was concretely the example here of MZ mine before Mascot server and then doing RT. So that is something he would never have considered himself, but could be worth exploring. But on the other hand, there are also workflow suggestions that are a bit less sensible. For example, domain experts can quickly say, okay, we started with data in term raw format. Compass export is suggested as a first tool of the workflow, but this tool can actually not work with term raw format. So that's wrong. What the problem there is that the annotation in bio.tools is wrong. So that annotation suggests that it can read these files, and Ape has no way to know that. That is more a question of how do we improve the annotations in the registry. Finally, in the full tool set with a bit even bigger set, we noted already that they are very similar to only the extended tool set, and the only notable difference is workflows that contain this tool here, term or raw file parser, which seems to be a rather new parsing tool, and therefore it has not yet been included in the proteomics domain. Now, once that happened, it would be in the other tool set and also be included in that. I mean, this is just some of the observation that we made on this, and of course, that needs to be in a more thorough way. But the preliminary conclusion that we draw from this is, so this Ape tool can be quite an effective means for searching for possible workflows in large collections of tools when they are semantically annotated, of course, that's a prerequisite. And not surprisingly, the quality of the obtained workflows highly depends on the quality of the semantic domain model, which in our case means EDAM and the annotations and bio.tools. So every improvement in this direction need to go together with the community developments that are happening there. And here already for this study, we evaluated the workflow suggestions manually and based on experience and some observations. But what is more desirable and what we will do in a next and follow-up study is to try to do that in a more objective way. Really not only taking workflow suggestions, but maybe just filtering them, but then also implement them and really benchmark them with reference data to see how they behave during execution. Because that is what we need to do eventually, but benchmarking all possible candidates from the outset is also not feasible because it's too time-consuming and computationally demanding. Right, so quick summary, take-home message kind of style. So we're working on a kind of route planner for scientific workflows. That is, I think, the one-sentence summary that I usually give to people when they ask what we're working on. We are getting better in both of the tooling and the results we're producing, but there's still work to do. So one thing is, of course, web interface for easier knowledge. We need to integrate that with workflow execution environments, benchmarking tools, sharing platforms like the Workflow Hub. We need to work a bit more on the algorithm to make it faster. So domain-specific heuristics is a term here that we're exploring. As I mentioned, we also need to take care of the quality of annotations in bio.tools, or if that's, realistically, we will never have only high quality annotations in the platform. So we need to conceive ways for filtering that so that we at least can correct it or only use those annotations that are of sufficient quality. And we need more, really evaluation of the whole procedure of exploration and the whole process on more real and realistic use cases. And that is my final sentence here, and call for collaboration. So I would be happy if you would get in touch if you think you have interesting workflow use cases where you would like to try out these technologies and help us developing it further. Because only by looking at real cases and getting real feedback on things that we not know before, we can really think what the crucial points are that we should prioritize and solve. So I would be happy if people would be interested to join in. A lot of potential for future projects and definitely for future papers, I think. Right, that was the update on our automated exploration work. Thanks for your attention. I see the chat active.
Thank you so much.
Anything
the chat,
in there?
no, it was not really related,
Okay.
not related questions. Thanks a lot.